are transported by the different electron transporters which are present in the inner membrane of the mitochondria to the molecular oxygen. Okay, and the molecular oxygen, after receiving the electrons from the reduced coenzymes, they are reduced to the water molecule. Okay, so in this process, so when this electron, a pair of the electrons from the reduced coenzyme passes through the complex 1, it results in the transporting of 4 protons from the matrix to intermembrane nucleus. Okay? So when the electrons move from the reduced coenzyme to the coenzyme Q through the complex 1, 4 protons are pumped out from the matrix to the intermembrane space. Okay? So this is one. Secondly, when this is wrong, actually this should go to the complex 3. Then oxygen, 10 numbers of the protons are transported across the mitochondrial membrane from matrix to the intermembrane space. Okay, so this is very important. Then some people, they are joining very late. Okay, now this is the component of the electron transporters. Okay, the components of the mitochondrial electron transport chain. So the first component we have mentioned that it is the NADH reductase. Now it has the molecular mass of 880, has 34 protein subunits. Then it has 2 prosthetic groups, that is the FMN and the adenine sulfur complex. Okay, then so this is the component. Then the complex 2, this I have not shown it earlier, that I have just shown the complex 1, complex 3 and the complex 4. So complex 3, it is the cytochrome reductase. Okay, the molecular mass is 250 and 10 subunits. Now it has 3 types of the heme, that is the heme B562, heme B566, heme C1 and the adenine sulfur complex, APS complex. Okay, then the complex 4 is the cytochrome oxidase. It has a mass of the 160, 10 protein subunits. Then it has the 2 hemes, that is heme A and A3. Then it also has copper, 2 copper, that is copper A and the copper B. Okay, succinate Q reductase. Now this is called as the complex 2, and this is required if the reduced coenzyme is FADH2. Okay, if the reduced coenzyme to be oxidized is FADH2, then the succinate Q reductase is also involved. Okay, so succinate Q reductase, so this is molecular mass is this one 140, it has 4 protein subunits. Then the phosphatic groups present are the FAD and the adenine sulfur complex. Okay, so these are the components of the electron transport chain. So here, the electrons from the NADH, we are starting with that how the NADH is oxidized. Okay? So electrons from the NADH enter the chain at the NADH2 reductase, and this is also called as the NADH dehydrogenase or the complex 1. Okay? So the initial step is the binding of the NADH and the transfer of its 2 high potential electrons to the flavin mononucleotide. See here. Now we have the FMN, which is in the oxidized form. Okay? So this is the structure of the FMN in the oxidized form. Now when it accepts 1 electron, okay? When it accepts 1 electron, then it is partially reduced. Okay? So here this hydrogen and the electron, it is attached to this nitrogen. Okay? So you have the partially reduced FMN. So this is called as the semiquinol intermediate. In the next step, another electron and a proton is accepted by the semiquinol intermediate. You have the reduced form. Okay, so that means the FMN, the first component of the NADH2 reductase, it accepts a pair of the electron, so that the FMN is completely reduced to form the FMNH2. Okay, so this is the initial step, the first step in the electron transport chain. Okay, same thing here. Just you have the NADH here, so the electron, a pair of the electron is transferred to the FMN. Now NADH is oxidized to the NAD plus, and the FMN is reduced to FMNH2. Okay, so this is the first step. In the second step, the electrons are then transferred from the FMNH2. So here, this FMNH2, it contains a pair of the electron. Now this is transferred to the oxidized iron sulfur complex. That is the second prostatic group of the NADH2 reductase. So this is accepted by the second prostatic group, that is iron sulfur complex. So when it accepts a pair of the electron, now it is reduced. Okay. So this is the reaction that is the portion which is shown in the green color. It is the component of the NADH2 reductase. Okay. So electrons in iron sulfur clusters with NADH2 reductase are then transferred or shuttled to coenzyme Q, also known as the VBC-028. Okay. So now we have the inner membrane of the molecule. It has the 10 isocyanate units. It's also called as the coenzyme 10. Okay. The flow of electrons from the NADH to QH2 through NADH2 reductase, it leads to the pumping of the 4 protons from the matrix to the cytosolic side of the inner membrane of the mitochondria. So when this pair of the electron, it passes through the complex 1, then it releases the energy. That energy is sufficient enough to push 4 protons from the mitochondrial matrix to the intermembrane space of the mitochondria. Okay. Now, if the electrons is to be oxidized or is to be transferred from the FADH2, then it passes through another protein complex, which we call it as the complex 2. Okay. So electrons from the FADH2 enters the electron transport chain at ubiquinone. Okay. It is coming to the ubiquinone. It doesn't pass through the first complex, that is the NADH2 reductase. It doesn't pass because the energy content of the FADH2 is less than the energy content of the NADH2 reductase. So it cannot pass electrons to the NADH2 reductase, but energy content of the coenzyme Q and the FADH2 is almost similar. So the electrons from the NADH2, it enters the electron transport chain to the coenzyme Q. Okay. So, and this enters through a complex 2, through a complex called as the complex 2, or it is also called as the succinate Q reductase. Okay. Succinate Q reductase complex and the other enzymes that transfers the electron from the FADH2 to the coenzyme Q are not proton pumps. Okay. So it doesn't pump the protons from the mitochondrial matrix to the intermembrane space because the free energy change of the catalyzed reaction is too small. Okay. So when the energy release is too small, it cannot push out the protons. Consequently, less ATP is formed from the oxidation of the FADH2 than from the NADH. So the electron from the coenzyme Q, the reduced coenzyme Q, that is the QH2, is transferred to cytochrome reductase, that is the complex 3. So the flow of the pair of the electrons through this complex leads in pumping of the 2 protons. Okay. So now this is the reaction mechanism that is occurring, how the electrons are transferred from the coenzyme Q to the cytochrome reductase. Okay. So it starts from here. Can you see this one? I think you can see this my cursor. Okay. So you have the QH2 in the red color here. Okay. So one electron from the QH2 is transferred to the iron sulfur complex. So this adenine sulfur complex, which is in the oxidized form, plus 3, it is reduced to adenine sulfur complex 2 plus. Okay. Then, so only one is transferred here, so that it forms the QH2 is converted to the semiquinone intermediate. Okay. Then, so this is the rest of the reaction. Now let's see here. Instead of seeing here, let's see here this figure. So you have the reduced coenzyme Q, that is QH2. The one electron is transferred to the adenine sulfur complex. Okay. Then to the cytochrome C1 and finally to the cytochrome C. So this, the adenine sulfur complex and the cytochrome C1, this is the prosthetic group of the cytochrome reductase. Okay. So first electron from the QH2 is transferred to the adenine sulfur complex. It goes to cytochrome C1, then finally the cytochrome C. So here 2 electrons are there. Only one is transferred. So the product generated here, it is the semiquinone intermediate. Now the electron from the semiquinone intermediate, that is, it has one more electron. So that is transferred to the Bl, okay, cytochrome Bl, then to the cytochrome BH, and finally that electron from the BH is transferred to a oxidized coenzyme Q. Okay, so that is converted to the semiquinone intermediate. Okay. So this is one cycle, or you can call it as the one part of the reaction. In the second part, another coenzyme Q, reduced one, is oxidized. So the first steps are same. One electron is transferred to the adenine sulfur complex to the C1 and cytochrome C, similar to the first part of the reaction. Okay. Then, similar to the above one, this is converted to the semiquinone intermediate. It has the one electron. So this electron is again transferred to the BL1, BH1, then finally to a semiquinone intermediate. In the first half, it is the oxidized coenzyme Q that accepts the electron. In the second half of the reaction, it is a semiquinone intermediate. Okay, so this will be reduced to coenzyme, with the reduced coenzyme Q. So that means if you sum up these two steps, then you will find that one reduced coenzyme is oxidized to the coenzyme Q, oxidized form of the coenzyme Q. So here, let's see the structure here. Okay, now this is the structure of the cytochrome reductase. So what it has as the prosthetic group, it has the adenine sulfur complex as the prosthetic group. It has the cytochrome C1 as the prosthetic group. It has cytochrome Bl. It has the cytochrome BH. Okay, so these are the prosthetic groups of the cytochrome reductase. So the electrons from the coenzyme Q, which is lying in the mitochondrial membrane, inner membrane of the mitochondria, it passes the first electron. It passes the adenine sulfur complex. Okay, then from the adenine sulfur complex, the electron is transferred to the cytochrome C1. From C1, this will go to the cytochrome C, which is somewhere here. Okay, it goes to that. So only one electron is transferred from the coenzyme Q, that is this reaction. So another one is with the coenzyme Q still in the form of the semiquinone intermediate. So the other electron now is transferred to Bl. Okay, then from Bl it goes to the BH. Then from the BH it transfers to a oxidized coenzyme Q. Okay, so this is the first reaction. In the second reaction, another coenzyme Q2 it enters it. First electron is transferred to the adenine sulfur complex. Second one, then it goes to C1, then it goes to cytochrome C. Okay, then the semiquinone intermediate. Yes, madam. It includes. Yesterday, Malaya, he sent it again. Yeah. No, I don't have. No, actually, what I know is Ganjana. I think she's not building that. Anyway, I can call. I'm having class now. After class, I'll call up. Okay, so like this, the coenzyme Q, the electrons, a pair of the electrons from the coenzyme Q is transferred to the cytochrome C, okay, through the cytochrome reductase. So this is the mechanism how the electrons are transferred from the coenzyme Q to the cytochrome C. Then finally, the electrons from the cytochrome C, it has to pass through the molecular oxygen through cytochrome oxidase. Okay, that is the complex 4. So cytochrome oxidase, that is the complex 4, it catalyzes the transfer of the electrons from the ferrocytochrome C, that is the reduced from the cytochrome C, to the molecular oxygen. That the molecular oxygen, it is the final acceptor of the molecular oxygen. So this is the reaction. So here, now to reduce the molecular oxygen completely, so molecular oxygen, we know that it has 2 atoms of the oxygen. Okay, so in water only one is there, H2O. So only 1 atom of the oxygen it goes. So that means these 2 atoms of the molecular oxygen, it has to be completely reduced. Otherwise, some other harmful products will be formed, like the superoxides. Okay, which are very harmful to the cell. Superoxides, it damages the biomolecules, especially the unsaturated fatty acids. Okay, so now this, to nullify that harmful effects of the formation of the superoxides, now this molecular oxygen, it has to be completely reduced. So the cell should have the mechanism that until and unless this molecular oxygen is completely reduced in the form of water, they should not be released from the complex. Otherwise, this is going to cause a problem. So this cytochrome oxidase digest the transfer of the electrons from the cytochrome C to the molecular oxygen in a such a way that this will release the molecular oxygen only when it is completely reduced to 2 molecules of the water. Okay. So here, 4 electrons are funneled to the molecular oxygen to completely reduce it to the water, 2 molecules of the water, and concomitantly pumps 4 protons from the matrix to the cytosolic site of the inner mitochondrial membrane. Okay, so this is the reaction mechanism, that how the electrons is transferred from the cytochrome C to the molecular oxygen. Okay, so what we have seen here, it is just the slide which I have shown in the previous slides. Okay, so here a pair of the electrons from the reduced coenzymes, if it is the NADH, then it passes its electrons to the coenzyme Q through the complex 1. Okay, so during the process, 4 protons are pumped out to the intermembrane space. Then the pair of the electrons from the coenzyme Q, it is transferred to the cytochrome C through the complex 3. Okay, in that process again, 2 protons are pumped out. Then when the electrons from the cytochrome C is transferred to the molecular oxygen through complex 4, 4 protons are again pumped out. Okay, so in total, 10 protons are pumped out when a pair of the electron, it passes from the reduced coenzymes or the NADH to the molecular oxygen. Now, for the electrons from the FADH2, it doesn't pass through the complex 1 because the energy level of the FADH2 is low. So it cannot pass the electrons to this. So the electrons from the FADH2, it passes to the coenzyme Q. It enters the electron transcription through the coenzyme Q. Okay, so one pair of the electron, it enters the coenzyme Q. Then from here it goes to cytochrome C through the complex 3. Then finally it will go to the molecular oxygen. So here, since the pair of the transfer of the electrons from the reduced coenzymes to the molecular oxygen, it is inhibited by some compounds. Okay, they are inhibited by some molecules or First one, that is the transfer of electrons from NADH2 reductase to the coenzyme Q. It is inhibited. Okay? It is inhibited by rotenol and the amytal. So rotenol and amytal, we know that these are the antibiotics. So these two antibiotics, it blocks the transfer of the electrons from the NADH2 reductase to the coenzyme Q. Similarly, the transfer of the electrons from the coenzyme Q to the cytochrome C1. Okay, cytochrome C1, we know that is the component of the cytochrome reductase. So it is blocked by antimycin. Okay. Again, another antibiotic. It is also blocked by the antimycin. Then finally, the final step, that is the transfer of electrons through the cytochrome oxidase. It is the carbon monoxide. Now this is the reason that why the cyanide, azide and carbon monoxide are highly toxic, highly poisonous to our system. This is just because that it blocks the final step of the oxidation of the reduced coenzyme. So if this is blocked, then of the ATP. So if the ATP is not produced, then definitely the cells cannot survive. Okay. So this is the reason that why the cyanide is poisonous to us. Okay, is highly poisonous to us. This is not required for you. This is just given you the structures of this one, that antibiotics. Okay. Now, what we have discussed till now is that how the electrons are transferred from the reduced coenzymes to the molecular oxygen so that the reduced coenzymes are oxidized. Okay. So here, the reduced coenzymes are oxidized by transferring the electrons to the molecular oxygen through a series of the electron carriers. Now, these electron carriers, it carries the electrons from the reduced coenzymes to the molecular oxygen. Okay. So since these complexes, it carries the electrons, they are called as the electron transporters. Okay. So here oxidation is through electron transport chain. So this is called as the electrons are carrying, so they are called as the electron transport chain. So we have not seen in any of these steps that how ATP is produced. Okay. But our topic which says that it is the electron transport chain and the oxidative phosphorylation. So what we have seen is simply the electron transport chain. We have not seen the oxidative phosphorylation. Okay. That is, oxidative phosphorylation means it is the production of the ATP when these reduced coenzymes are oxidized. So how it is oxidized, we have seen it. How the ATP is formed, we have not seen it. Okay. So this electron transport chain and the oxidative phosphorylation, these are coupled reaction. It is a link one. If one is not happening, the other one will not happen. Okay. If there is no electron transport chain, there will not be formation of the ATP. The oxidative phosphorylation would not occur. If the oxidative phosphorylation is not occurring, then electron transport chain will not occur. So this is a coupled reaction. It is a linked reaction. So now the question is that how does the oxidation of the reduced coenzymes, it leads to the formation of the ATP? So there was many hypotheses which tries to explain that how the ATP is formed when the coenzymes are oxidized, when the reduced coenzymes are oxidized. Okay. So one is the conformational change hypothesis. So according to that, it says that when these reduced coenzymes are oxidized, energy is liberated. That energy is absorbed by some biomolecules like the protein. Okay. And because of the absorption of the energy, the protein molecule, it changes the conformation. Okay. Then when the protein it returns back to its native conformation, then that energy is released, that is utilized by the cells to form the ATP from ATP and the inorganic phosphate. Okay. So since there is a change in the conformation of the protein, that hypothesis is called as the conformational change hypothesis. Okay. Now this was not experimentally proved. Okay. There was no experimental evidence to prove that the ATP formation cases were there which was not supported by the by any evidence. Okay. So finally, Peter Mitchell in 1961 he proposed the chemiosmotic hypothesis. Okay. So here oxidation of the reduced coenzyme is linked to the formation of the ATP. Okay. So he proposed that or he explains the formation of the ATP during the oxidation of the NADH or the FADH2. So what he says is that the transfer of the electrons through the respiratory chain leads to the pumping of the protons from the matrix to the form the ATP. See here. Now you have the mitochondrial matrix, okay? You have the intermembrane space. So when the electrons are transferring through this inner membrane of the mitochondria, then that results in the pumping of the protons. These protons from the matrix it comes out to the intermembrane space. So this the concentration of the proton in the intermembrane. The same slide also, the next slide also shows the same thing. Okay. So here we have the complex 1, complex 3, complex 4. Okay. Then complex 5. We have not discussed about the complex 5. So here the electrons, okay, the electrons are transferring from the reduced coenzymes through these complexes finally to the molecular oxygen. So during this process, when the electrons moves through these complexes, then it pumps out the proton. Okay? The protons are coming out from the matrix to the intermembrane space. So because of this, the concentration of the proton in the intermembrane space will be high. Here, the concentration will be low. Okay? So there is a difference in the potential of the proton. So there is a electrochemical gradient is generated. Okay? Now, when this hydrogen ion or the proton in the intermembrane space, when it comes back to the matrix, because here the concentration is high, is low, so there will be definitely the protons there will have the tendency to move from the higher concentration towards the lower concentration. Okay, but we know that the inner membrane of the mitochondria is impermeable to protons. Okay, so it is highly impermeable to the proton. So these protons from the intermembrane space, it cannot pass through the membrane. Okay? It cannot pass through the membrane. So they cannot come through this. Okay? So that is the reason that why there is a difference in the concentration. Now these protons from the intermembrane space, it can come inside the matrix through a protein complex. Okay? So that complex we call it as the complex 5, or later on we will also be calling it as the F1 ATPase. A protein complex, for time being, we just remember that it is the complex 5. So these protons from the intermembrane space, it comes back to the matrix through the complex 5. Okay. So when this proton it moves through the complex 5, this complex 5 it catalyzes the phosphorylation of the ATP and the inorganic phosphate. So this inorganic phosphate is linked to the ATP to form the ATP. So ATP is generated. So how this ATP formation is possible? It is just very clear to you. Do you have any questions in this part? Yes, any question in this part?
No, sir.
No question. Let me stop for a few minutes. I'll just take your attendance. No? We'll continue after I take the attendance. I will continue it. So, row number 1 is there.